Let's look at this A-level biology question. So, can you explain 3 ways that the structure of a fish gill is adapted for gas exchange? Okay, let's go through 3 possible answers. So, the first thing we can say to get a mark is fish gills have many filaments, or same marking point, but you can say many lamellae. Now remember, this is an explain question, so you can't literally just give the structure. You can say many filaments or many lamellae to increase the surface area, so faster or more efficient gas exchange. Okay, so if you remember you're maybe doing your fish gill dissection, as I'm sure lots of you have, when you look at the gills, you'll see lots of gill filaments, and on each gill filament, you've got loads of folds, loads of lamellae. So collectively together, you've got a huge surface area. And as you know, having a large surface area is one of the key adaptations for any surface that is adapted for gas exchange. Second thing we can say, the lamellae are very thin, okay? Now, explanation-wise, we're going to explain that by saying this gives a short diffusion pathway, which again means faster diffusion or gas exchange. Okay, remember in the lamellae are the capillaries, the teeny tiny blood vessels, and the oxygen is in the water, and the oxygen has to diffuse from the water into the blood. So by having thin lamellae, it means that the oxygen does not have to diffuse very far at all. In fact, it only has to cross a couple of layers of cells and it can get directly into the bloodstream. So it's much faster. Third, and probably the most complex idea to explain, is that the blood in the capillaries and the water flow in opposite directions. Now, this is known as the counter-current principle. In fact, you could just say the counter-current principle, or you could say the blood and the water flow in opposite directions. Now, explanation-wise, why is that a benefit in terms of gas exchange? Well, it means the diffusion gradient for oxygen is maintained along the whole length of the gill. Okay, so the whole length of the gill, there will always be a diffusion gradient between the oxygen in the water and the oxygen in the blood, which basically means diffusion will continuously occur. Oxygen will keep diffusing from the water into the blood along the whole length of the gill, which is a great adaptation for gas exchange. Let's have a look at the Krebs cycle. Now, I know this is something that you guys wanted to go over in these videos, and don't forget to click the link in my bio if you do want to join my A-level biology course. Now, the Krebs cycle is the 3rd stage of aerobic respiration, and we've got a simplified diagram here where basically they are asking us to complete the carbon numbers in the compounds. So let's start with this side. We've got something combining with an acetyl group. Now that acetyl group, don't forget, comes from the acetyl coenzyme A that is produced at the end of the link reaction. Okay, so the link reaction is stage 2. It produces acetyl coenzyme A, and the acetyl group will feed into the Krebs cycle in the mitochondrial matrix. Now, the acetyl group has 2 carbons. Okay, we know that. This compound here has 4 carbons. if you're doing OCR, you do actually need to know the name of that compound. Okay, it's called oxaloacetate and it has 4 carbons, which is what the question wanted us to do. So we've got a 4-carbon compound joining with the acetyl group, which has 2 carbons. That will give us a 6-carbon compound. Now again, if you're doing OCR, then the name of that 6-carbon compound is actually citrate or citric acid. Now they've given us a bit of help with the next part of the diagram because you can see that citrate is being decarboxylated. Decarboxylated just means CO2 has been removed, but they've helped us a little bit there because if we remove carbon dioxide from a 6-carbon compound, it's obviously losing 1 carbon. So this must be a 5-carbon compound, which makes sense because it loses another CO2 here, or it's decarboxylated again, and then we've gone down to a 4-carbon compound. The whole point of this, by the way, well, there's a few reasons, but we're trying to regenerate the 4-carbon compound so the cycle can continue. But we are also going to make some important products in the Krebs cycle. So let's have a look at this alternative question. Can we name 3 other substances produced by the Krebs cycle? On here, it's really only showing CO2, but other important substances that we will make per cycle, we will make 1 molecule of ATP. We will also make 3 molecules of NADH, or you can call that reduced NAD, and 1 molecule of FADH, or you can call that reduced FAD. Now the ATP, I know it doesn't seem like a lot, just 1 ATP, but the ATP here is produced by substrate level phosphorylation, which means there's a donor molecule donating the phosphate to ADP to make ATP. The coenzymes are really useful because our NADHs and FADHs are going to go to stage 4 of respiration, which happens on the cristae, and they are going to allow us to make loads more ATP. Let's have a look at this exam-style A-level biology question where we're going to be labelling the main chambers of the heart as well as the main blood vessels in the circulatory system. Let's start with the heart itself. Now you can see on the diagram we've split the heart into 4 chambers. So you should be able to name A as the right atrium and B as the left atrium. Now, initially when you look at it on the paper, you might think, oh, hang on, that's the wrong way around. But remember, we're imagining that heart is inside our chest. So the kind of right and left is flipped as to how you would see it initially on the paper. So do be careful with that. C is obviously the right ventricle. And D is the left ventricle. And you might be able to recognise the left ventricle more easily. If it's a more realistic diagram, you might be able to see that the wall of the left ventricle is thicker. We can't see that here, but on some diagrams they have shown you that as well. Let's go ahead and label the main blood vessels in our circulatory system. So let's start with E. E is going from the right ventricle to the lungs. So that is our pulmonary artery. Remember, I know it's an artery because arteries always go away from the heart. This one's going to the lungs, so it's the pulmonary artery. This one here, F, is the pulmonary vein, and it's going in to the left atrium, and I know it's a vein because veins go in to the heart. If we move down to G, G is our aorta, which is the main artery in the body, which leaves the left ventricle and pumps blood around the rest of the body. H is going to the kidney. So this is one that you don't have to know at GCSE, but you will now have to learn at A level. The blood vessel that goes to the kidneys is called the renal artery. And again, I know it's an artery because it's going away from the heart to the kidney. J is the renal vein. It's going from the kidney back in to the heart. All we've got left to do then is I, which is our vena cava, which is obviously a vein because it's going into the heart. It's bringing blood back from the rest of the body back into the right atrium. So here is a scientific drawing of a mitochondrion. Now you might be thinking it doesn't look great, but that's the point. The question is asking us, how could we improve the scientific drawing? For 2 marks, we need 2 suggestions. So there's quite a lot of ways we could make this better. I've made it particularly bad. First thing I notice immediately is there should be no colouring or shading on a scientific drawing. Remember, this is not art, this is biology. So we shouldn't be shading and we shouldn't be colouring in. Don't get your coloured pencil crayons or your felt tips out. Should not be colouring in our biological drawings. Number 2, lines should be continuous. Or you could say no sketchy lines, no hanging lines. Now, what I mean by that, I've tried to show you some sketchy lines here. So I've done a little bit of feathering or sketching. That is not okay. I've also done some kind of overlapping or disconnected or hanging lines, which is also not okay. All the lines should be clear, crisp, sharp, and connected. Something else that I could say, I should include a magnification or a scale bar. So if you see a biological drawing in the exam and they haven't told you what magnification was used, or they haven't included a scale bar so you can work it out, that is definitely something that you should suggest, because you should always have that on a biological drawing. And finally, add labels. A biological drawing is no good without labels, so you should use a ruler, nice straight label lines, and label the different features of the mitochondrion, like the matrix, the cristae, and the outer membrane. Right, let's have a look at this A-level biology question. It's about co-transport, which is a topic that lots of students can find tricky. So it's asking us to explain how glucose is absorbed by the cells lining the ileum into the blood. Now, the ileum is part of your small intestine. It's where absorption takes place. So it's where glucose leaves the small intestine and is absorbed into the bloodstream. So it uses co-transport. Let's make a start on answering the question. First of all, we're going to say sodium ions are actively transported into the blood. Okay, so let's get that on the diagram to help you understand. So this is one of the epithelial cells lining the ileum. You can see it's got microvilli, as a lot of cells adapted for absorption do. It's all about increasing the surface area. But this cell is going to actively transport sodium ions out of the epithelial cell and into the blood. So this is my capillary here, if you weren't sure, and the sodium ions are being actively transported out of the cell into the blood. Now, what's the point in that? Marking point 2, this creates a diffusion gradient for sodium ions. Or another way to say that is it maintains a low concentration of sodium ions in the cell. Okay, so by continuously pumping them out of the cell, you're going to maintain a low concentration of sodium ions in the cell, or you're going to maintain a diffusion gradient for sodium ions. Basically, what that will cause to happen is sodium ions to start diffusing into the cell because you've got a low concentration in the cell, a higher concentration in the lumen, the inside of the small intestine. So sodium ions will start to diffuse in. But this is where co-transport comes into play. As the sodium ions diffuse in, they bring glucose with them using a co-transport protein. Okay, let's add it onto the diagram. So the sodium ions are diffusing in, but this is a special type of protein called a co-transport proteins. So as the sodium ions diffuse in, they bring in with them a molecule of glucose. So we can call that co-transport a co-transport protein. It's also an example of facilitated diffusion because the sodium ions are diffusing in using a transport protein. So you can call it co-transport or facilitated diffusion. Now that co-transport didn't need energy from ATP, but the active transport did. So the active transport requires energy from ATP because it's moving it against its concentration gradient. And then by creating a concentration gradient for sodium, sodium ions and glucose can then diffuse in using co-transport. Finally, once the glucose is in the epithelial cell, the glucose can then diffuse into the bloodstream. So I'm going to put that here. Now, obviously, this is not simple diffusion because we're talking about glucose here. So it will be facilitated diffusion. It will need a transport protein, a carrier protein. But because you've brought the glucose into the cell, you've now got a high concentration of glucose in the cell, so glucose can diffuse into the blood. And there you go, we have absorbed the glucose from inside the lumen of the small intestine, across the epithelial cell, into the bloodstream, with the help of our sodium ions, and that is what we call co-transport. Okay, let's have a look at this maths question that could come up in biology A-level. So we've got an experiment about respiration in yeast, and we've been told that the yeast cells respired and produced carbon dioxide, which was collected in a gas syringe. 50 centimetres cubed of carbon dioxide was collected. Now the gas syringe measures to the nearest centimetre cubed, and they're asking us to calculate the percentage error in this measurement. Now, percentage error, let's just remember the kind of equation or the way in which we work it out. To get percentage error, we need to do the uncertainty of the measurement divided by the amount measured, and then we multiply it by 100. So first of all, what do we mean by uncertainty? Well, every piece of apparatus that you use to measure something will have a degree of uncertainty. Like you can't be 100% sure that you have measured that volume accurately. So here we've measured 50 centimetres cubed using a gas syringe. The gas syringe measures to the nearest centimetre cubed. Now the uncertainty is always half of the smallest division. So you look at the apparatus, or you get told about the apparatus, and you figure out, well, what is the smallest division, because the uncertainty will be half of that. Now here we can't see the gas syringe, but we're told it measures to the nearest 1 centimetre cubed. So the uncertainty in our measurement will be half of that. Okay, so we say that the uncertainty is half a centimetre cubed. Basically what that means is when you've measured your 50 centimetres cubed, in reality, it might be half a centimetre cubed more than that or half a centimetre cubed less than that, which is why we put plus or minus before our uncertainty, okay? Once you've figured out what the uncertainty is by halving the smallest division on the apparatus, you can then go back to the equation. So you just do the uncertainty, which for us was 0.5 centimetres cubed. You divide it by the amount you've measured. So in this case, we'd measured a volume, and the volume we'd measured was 50. And then you times that by 100. So 0.5 over 50 is 0.01. Obviously, if we times that by 100, the percentage error in our measurement is 1%. Can you describe how a phagocyte can destroy a pathogen once it has entered our bloodstream? Now, this is a really nice kind of storytelling question, and we're going to do some diagrams as well to help you understand it a little bit more. First thing, marking point number 1 is obviously the phagocytes, which, don't forget, is a type of white blood cell. The phagocyte will engulf the pathogen. So let's show that on a little diagram. Here's our phagocyte. Here's our pathogen. What happens is the cell surface membrane of the phagocyte basically wraps itself around the pathogen, and that's what we mean by engulfs the pathogen. If you can imagine, once it's kind of brought that pathogen in, it's like endocytosis, isn't it? It's the same idea, endocytosis. Once it's brought that pathogen in, it's going to wrap that membrane around it and enclose it within a vesicle. So marking point 2, we can say that the pathogen is enclosed in a vesicle, but that vesicle does have a specific name, which, you know, it's not hard to learn, so I would use it. We call it a phagosome. Okay, so if I just edit my diagram, if I move it on a little bit, that's basically what will happen. So the pathogen is now inside the vesicle, which we call the phagosome. Now, the next thing to happen is lysosomes that are inside the phagocyte fuse with the phagosome, and hydrolytic or digestive enzymes hydrolyze or digest the pathogen. Okay, so let's just show that on the diagram. So here's my little lysosome over here. Could draw a few of them on there. So my lysosome is going to fuse with my phagosome, kind of like this. It's going to release its digestive enzymes into there, and those enzymes are going to hydrolyze or digest, which just means break down the pathogen. Another word that you can use if you want to be really fancy, you can call the vesicle a phagosome. When the lysosome fuses with the phagosome at this point, you can call it a phagolysosome. You don't have to, but you're basically just shoving those words together. Phagosome, lysosome, you've now made a phagolysosome, and it's at that point that the hydrolytic enzymes will digest and break down the pathogen. Let's have a look at this practical question from A-level biology. Now, the question is asking us, during the experiment, the coloured liquid is moving towards the right. Can we explain why? Now, if you look at the apparatus carefully, this is probably similar to something you've already seen. It's very similar to a respirometer, which you may have studied, but there is a substance in this conical flask that absorbs oxygen. Now, normally what we see is a substance that absorbs carbon dioxide, but they're trying to make this a little bit more challenging. So, if there's a substance in here that absorbs oxygen, the yeast, which has been provided with glucose, is going to be respiring anaerobically because there's going to be no oxygen for respiration in that conical flask. So that's going to be marking point 1. Yeast respires anaerobically. because all of the oxygen is being absorbed by this substance. Now, anaerobic respiration in yeast, you should know, produces ethanol and carbon dioxide. So CO2 is produced. I'm also just going to make a point of saying that the oxygen is absorbed. Okay, so the oxygen is being absorbed by the alkaline pyrogallol. The yeast is respiring anaerobically because there's no oxygen. That produces carbon dioxide. And now we should be able to explain why the coloured liquid moves to the right in this glass tubing, this capillary tubing that we've got attached at the top. So the reason it's going to move to the right, if you think about it, the CO2 that's being produced is going to increase the volume of gas in the tubing, or you could say increase the pressure in the glass tubing, which is basically going to push that coloured liquid away from the flask and to the right, because there's an increase in volume of gas here and an increase in pressure caused by the production of that carbon dioxide. It's actually not a bad question. It's just really important that you look at the diagram and realise that all of the oxygen is being absorbed. Can you explain the advantage of the Bohr effect during exercise? Now, the Bohr effect is referring to the effect of carbon dioxide on hemoglobin in the blood. Obviously, when we exercise, our muscle cells are doing more aerobic respiration and producing more carbon dioxide. And that carbon dioxide is going to cause what we call the Bohr effect and affect the structure of hemoglobin. In fact, just to explain that a little bit further, the increase in CO2 is going to lower the pH of the blood, and that will affect the overall tertiary and quaternary structure of the hemoglobin protein. So, why is that an advantage during exercise? Well, first of all, the change to the tertiary structure of hemoglobin, the hemoglobin's affinity for oxygen is going to be reduced. So it's going to reduce hemoglobin's affinity for oxygen. Remember, affinity is kind of how easily the hemoglobin loads with oxygen or associates with oxygen. And in increased CO2 levels, we're going to reduce hemoglobin's affinity for oxygen. So what that means is hemoglobin will more readily, or you can say more easily, dissociate with oxygen, or the alternative to that is unload oxygen. Okay, so because it's got a lower affinity for oxygen, it will more readily dissociate or unload with oxygen. I kind of picture that as like it's more eager to give the oxygen away, if you like. Now, what's the advantage of that? It means during exercise, the muscle cells get more oxygen for aerobic respiration, which obviously they're going to need to contract. We know that muscle contraction requires a lot of ATP, so they're going to have to do a lot of respiration, and this is going to allow them to get plenty of oxygen to keep doing aerobic respiration. The other idea that you could get across to get the third mark is it will delay anaerobic respiration, or you could say less lactic acid produced, okay? Because if the hemoglobin is unloading more oxygen and the muscle cells keep getting oxygen for longer, they can keep going with their aerobic respiration and delay the anaerobic respiration, which obviously produces lactic acid, which causes muscle fatigue. It's all about the keywords with this one, using the correct terminology, so using words like affinity for oxygen, dissociate or unload, and then obviously remember to stress it's aerobic respiration here because we're talking about oxygen being available for aerobic respiration. Can you explain how a single base substitution can cause a change in the overall structure of a polypeptide? Let's just remind ourselves what a single base substitution is. You're basically being told that one of the bases has been switched or substituted for another base. So maybe the base was meant to be adenine, but now instead you've got guanine, for example. Now, if you change a base like that, you are changing the triplet in the DNA, and then you are changing the codon in the mRNA. So ultimately that could have an effect on the final polypeptide. Let's go through 3 marking points to explain that. So if you change a base in the DNA or the mRNA, that could potentially alter the order of amino acids or the primary structure of the polypeptide. Okay, because you've switched one base, so the triplet is different or the codon is different, and it could possibly code for a different amino acid. So you've basically changed the order of amino acids. That's not always the case, is it? Some of these mutations are silent because the genetic code is degenerate. So it might be that even though you've switched one base, it actually still codes for the same amino acid. But we're not going to go down that route with this question because it's telling us it causes a change in the polypeptide. So we're going to assume that it has altered the order of amino acids or the primary structure. Now, to continue with that, for it to affect the overall shape of the protein, it must affect the bonds that hold the tertiary structure together. So those bonds are hydrogen bonds, ionic bonds, disulfide bonds. You only have to name one of them, by the way, but you do have to name one. You can't just say the bonds. So you could say the ionic bonds or the disulfide bonds or the hydrogen bonds. They would form in different places because obviously if you've got a different order of amino acids, it's going to affect those bond formations in the tertiary structure. So finally, you'll end up with a different tertiary structure, or a tertiary structure that is a different shape. Now, I'm just going to stop there because I don't know what kind of protein this is. It just says polypeptide. Obviously, if the question was telling me it was an enzyme, I could go on to say that the active site would be a different shape and it wouldn't be complementary to the substrate, and we wouldn't be able to form enzyme substrate complexes. But I don't know that this is an enzyme. It could be another kind of protein, it could be a receptor, it could be a hormone or whatever. So I'm just going to leave it with changing the structure of the polypeptide because you've got a change or a different tertiary structure. Now that's quite a standard answer for this kind of question. It's definitely something that you should be able to just quickly write down and fully understand. Right, let's describe how alterations in tumour suppressor genes can lead to the development of tumours. Now, a tumour is basically an abnormal mass of cells, and that has formed due to uncontrollable cell division. Now, that is usually the result of a mutation in either a tumour suppressor gene or a proto-oncogene. This question is just asking us about the tumour suppressor genes. So, first marking point is for simply saying that it could result from mutations in the tumour suppressor gene, which I'm just going to shorten to save time. So, mutations in the tumour suppressor gene. Obviously, what that could lead to is a change in the order of amino acids in the protein. Okay, now remember, the tumor suppressor gene codes for proteins that regulate and slow down the cell cycle. Now, if there's a mutation in the tumor suppressor gene, then the proteins that are coded for by that gene will have or could have a different order of amino acids, which then means the tertiary structure of the protein could be altered. But remember what this protein does. This protein regulates and slows down the cell cycle. So if we're changing the order of amino acids in that protein as a result of a mutation, and we're changing the tertiary structure of that protein, it will be non-functional, which means it won't be able to regulate or slow down the cell cycle. Hence why we can get uncontrollable cell division and the formation of a tumour if there's a mutation in that tumour suppressor gene. Something else that we can suggest is increased methylation of the tumour suppressor gene, or this is known as hypermethylation. Now, if you get increased methylation, that is going to prevent that gene from being transcribed, because you can get increased methylation of the cytosine bases in the promoter region. And if the promoter region of the gene, of the tumor suppressor gene, is hypermethylated, it basically means that transcription is blocked. That gene cannot be expressed. Can't be transcribed, can't be translated, you can't make the proteins. So again, you're not making the proteins that should be regulating the cell cycle, and that's why it can lead to uncontrollable cell division and the growth of a tumour. Right, we're going to do a genetic cross, an A-level genetic cross, or Punnett square. The way I would approach these questions is I'd read it carefully, because they're going to give you a lot of information in the question. And I would think to myself, right, what kind of cross is this? Because there are loads of different ones we have to learn. Now, as I read through this question, they're telling me about two different genes. They're telling me about the tall and dwarf alleles, which are one gene, and then they're telling me about colour as well. So I've got red allele and white allele. So I know immediately this is going to be a dihybrid cross, because I'm looking at the inheritance of two traits simultaneously. The other thing that I notice is the alleles for flower colour. Now, these are obviously codominant alleles because we've used a different representation for those alleles. We've used the letter C for colour to illustrate that they are alleles of the same gene. And then we've used the capital superscript letters of R and W, both capitals, to show that they are both codominant. We can't use an uppercase and a lowercase letter if they're codominant alleles, because one is not dominant over the other. In fact, if both of those alleles are inherited, the offspring would express both colours and therefore appear pink in this case, right? So I've got a dihybrid cross, I've got codominance. Let's go, let's see if we can do it. We're going to cross a dwarf pink plant. Now, dwarf, I'm told, is recessive. So to be dwarf, that parent plant must have two recessive dwarf alleles. They're also pink, which tells me they've got a red and a white allele for colour. So they're heterozygous for that one, which is why they've got that intermediate phenotype of pink colour. I'm crossing them with a heterozygous tall white plant. So if they're white, they've got two of the white colour alleles. Okay, I'm crossing them together. That's going to get you marking point 1. You've correctly figured out the parental genotypes. So let's go ahead and do the genetic cross. Now, in terms of the gametes that this parent could produce, this allele could combine with that allele, or this allele could combine with this allele. Now, I could repeat it, but I can see that if this allele combines with this allele, I would get the same combination. And if this allele combines with this allele, I would get the same combination. So there's actually only 2 different gametes that parent can produce because they are homozygous for that first gene. So there's only 2 different gametes. This parent, let's do the same. So when the gametes are produced in meiosis, we could get this combination. I'm not going to repeat that because I can see that that and that would give me the same combination. Or we could get this combination. And again, I'm not going to write that down again because I see that that and that would give me the same. So again, there's only 2 gametes this parent can produce, which makes sense because they are homozygous for that white colour allele. Now I've got the gametes, I can do my genetic cross. I'm going to write this parent's gametes over on this side. So I can do my little grid, make my life a little bit easier. And then I'm going to show the combinations at fertilization. So remember at this point we're saying, well, this gamete could fuse with this gamete, or this gamete could fuse with this gamete, and we're just filling in the combinations. So let's get them all in. This one. So I'm just putting the alleles back together in their pairs. Okay, now you're going to get another mark if you correctly get those offspring genotypes. Now, let's go through the offspring and make a note of the phenotypes, because that's what's going to get us marking point 3. So this plant is going to look, I'm just going to write it underneath, tall, because it's got the dominant allele, and it's also going to look pink because it's got a red and a white. This offspring is going to look tall because it's got the dominant tall allele, and it's going to look white because it's got 2 recessive white alleles. This offspring is going to be dwarf. It's got 2 recessive dwarf alleles, and it's going to be pink. This offspring will also be, rub that out, spelt dwarf wrong. This offspring will also be dwarf and white. So marking point 3 is for correctly linking the offspring phenotypes. Finally, I'm going to give a ratio. So they're all different. Look, tall pink, tall white, dwarf, white. They're all different. So the final ratio is a 1:1 to 1:1 ratio. And that's it. It's dead easy when you know how to do it. It's just like a little puzzle, isn't it, that you've got to solve. So the more you practice, the better you will get. Just make sure you read the question really carefully, you know what kind of genetic cross it is, and that you get those parental genotypes correct, because then you're going to be able to do the rest, no problem. Let's go ahead and explain why oxygen is needed for the production of ATP on the cristae of the mitochondria. Now, if you haven't realised, this question is about oxidative phosphorylation, which is the final stage of aerobic respiration. I think we've already done videos on glycolysis and the Krebs cycle, so this needs to go with those videos. This is the final stage, oxidative phosphorylation, which means making ATP in the presence of oxygen on the cristae. Why is oxygen so important? The first thing we're going to say, we always say this, is that oxygen acts as the final electron acceptor. Really important marking point. Just write that down straight away as soon as you see a question about oxygen and oxidative phosphorylation. So it acts as the final electron acceptor. What do we mean by that? Electron acceptor. Well, remember, electrons pass down the electron transport chain. And it's that transfer of electrons that releases energy in order to make ATP from ADP and PI. Whenever I say make ATP, I always put from ADP and PI, just because sometimes they don't give that mark unless you say what it's made from. So oxygen is the final electron acceptor. It's accepting the electrons that have passed down the electron transport chain. As those electrons pass down the electron transport chain, that is providing the energy that is ultimately going to be used to make ATP from ADP and PI. Something else we can say is that the oxygen, when it accepts the electrons, and don't forget, it also accepts protons from the NADH or the FADH. So the oxygen, when it acts as the final electron acceptor, and it also accepts those protons, it will form water. Okay, so oxygen forms water. At the end of the process, it's going to accept the electrons that have just passed along the ETC. It's going to accept the protons that originally came from NADH, FADH, and that acceptance of protons and electrons, basically hydrogen, is going to form water, which, as we know, is one of the waste products of aerobic respiration. Now, these are the marking points for this question. I have simplified it here. Like if this was a 5 or 6 mark question, don't forget that you should be able to talk about the formation of the proton gradient, which I've missed out in this video. But basically those electrons being passed down that chain provides the energy to form that proton gradient, and it's the proton gradient that drives the production of ATP. I'll try and do another video about chemiosmosis to explain that in a little bit more detail. So let's talk about adrenaline. Now, adrenaline, as you know, is a hormone secreted by your adrenal glands, and it's involved in what we call the fight or flight response. Now, with this question, they're telling us it binds to receptors on the plasma membrane of liver cells, and when it does so, it leads to an increase in blood glucose concentration in the same way that the hormone glucagon does. So can we explain what is happening here? Well, first of all, just remember, adrenaline is a hormone made out of protein, so it doesn't actually enter the cell. It's not going into the liver cell, it just binds to its receptor that is on the cell surface membrane, and then it leads to a kind of cascade of reactions within the cell that will ultimately help to increase the blood glucose level. So let's go through that cascade of reactions. Now, you should know this as the second messenger model. So first thing that's going to happen, when adrenaline binds to its specific receptor, it's going to activate an enzyme called adenylate cyclase. And you do need to know the name of that enzyme. So it activates that enzyme within the liver cell. Now, what that enzyme is responsible for, so adenylate cyclase will convert ATP into cyclic AMP. And cyclic AMP is what we call the second messenger. So as an alternative to marking point 2, you can say that a second messenger is produced, but you could be asked to name it. So make sure you know the second messenger is cyclic AMP. The first messenger, by the way, is adrenaline. That's acting as the first messenger, but it's not going into the cell. It's activating the enzyme adenylate cyclase, which then leads to the production of the second messenger inside the cell. Let's continue. What does cyclic AMP do? It activates another enzyme, which is called protein kinase A, or pKa for short, and it's that enzyme that will ultimately lead to an increase in blood glucose levels, because this enzyme stimulates glycogenolysis as well as gluconeogenesis. Remember, glycogenolysis, if you look at the words, lysis means splitting or breaking down. So what we're saying is glycogen is being broken down back into glucose. Gluconeogenesis, we're saying we're making Genesis new, which is neo, and glucose. So this is making new glucose from other sources, such as amino acids or glycerol. Both of those things are what is going to lead to that increase in blood glucose, because both of these processes will result in more glucose diffusing back into the bloodstream. Ecology. Let's have a look at an A-level biology ecology question. We've got describe and explain how succession occurs. Right, so first thing we're going to say, when you have primary succession, remember you're starting with bare rock, like land that has never had anything growing on it before. We're going to start with our pioneer species, and it's going to colonize the bare rock. So marking point 1 is really for that idea of colonization by what we call the pioneer species, the first species to grow in that area or habitat. Now, what that will do is change the abiotic conditions in that area. Or you can say it will change the environmental conditions in the habitat. Because those first species, when they die and are decayed, you'll start to get like a fine layer of the first soil in that area. Ultimately, what this is doing by changing the abiotic conditions, it's making the environment less hostile or less harsh. So then, new species will grow, and they will outcompete the pioneer species. And the same process really repeats over and over again. So new species will grow, they will continue to change the abiotic environment, they will continue to make the conditions less hostile, and you'll go through this succession where you kind of start off with lichens, and then you get mosses, and then you get some grasses and shrubs, and then some flowering plants, and then some small trees. Finally, you're going to get your big trees. So let's think of our final marking points. As this process continues, you get an increase in biodiversity. So the area is becoming more and more diverse. You also get an increase in biomass as more and more living things are growing there. And ultimately, you will reach a stable climax community, where you'll see little change over time. So the populations will now remain stable at their carrying capacity, and the abiotic conditions will stay pretty stable too. That's what we call the climax community, which in the UK is woodland. Right, let's look at a question about the eye, specifically about receptors in the eye on the retina. So you'll know that you have cone cells and rod cells. They're both types of photoreceptor in the retina. Cone cells give higher visual acuity than rod cells. Explain how. Visual acuity is that idea of seeing things in more detail. They allow us to see things more clearly. Like even if two objects are very close together, we'll be able to see and distinguish them as two separate objects because the cone cells give this high visual acuity. I suppose it's a bit like a microscope when you say it has high resolution. Except don't confuse the two things, okay? We need to say high visual acuity, not high resolution. So why do they have high visual acuity? Well, cone cells, if we start with that, do not show retinal convergence. Rod cells, on the other hand, do. That's why they have low visual acuity. Okay, so that's a key word that's going to be really useful for you to remember. Cone cells do not show retinal convergence. What that means is each cone cell synapses with a single bipolar neuron. Okay, so there's no summation, if you like. Whereas with the rod cells, you will have several rod cells all synapsing with the same bipolar neuron. With the cone cells, that's not the case. Each cone cell is connected to or synapses with one separate bipolar neuron. Now, what that ultimately means is that you will get a separate impulse going to the brain. Remember, the bipolar neurons synapse with the optic nerve, and that's how the electrical impulse gets from the receptor to the brain via the optic nerve. But with your cone cells, each cone cell synapses with a single bipolar neuron, so you'll get separate impulses going from each cone cell to the brain, which is why you will perceive objects as separate objects, even if they are close together. Rod cells, remember, is just the opposite. Rod cells do show retinal convergence. They have low visual acuity because you get several rod cells synapsing with one bipolar neuron, so you only get one impulse going to the brain. Right, let's look at this A-level biology question about hemoglobin. So the question is asking us to explain why the binding of one molecule of oxygen then makes it easier for the second molecule of oxygen to bind. So the way this works, and for marking point 1, we're going to say that when the first molecule of oxygen binds, it actually alters the tertiary structure or the quaternary structure of the hemoglobin. So when one oxygen molecule binds, it alters the tertiary, or you can say the quaternary structure of hemoglobin. But don't just say the shape of hemoglobin. Remember, hemoglobin is a protein. It has a primary, secondary, tertiary and quaternary structure. And when that first molecule of oxygen binds, it will change the tertiary or the quaternary structure, which is what makes it easier for the second molecule of oxygen to bind. And the reason it's easier for the second molecule of oxygen to bind is that when this happens, it uncovers or exposes a second binding site, or you can just say another binding site. Another way to get this mark, by the way, is to say that it uncovers or exposes another heme group. Because you may remember that in hemoglobin there are 4 polypeptide chains, and in each polypeptide chain is a heme group, and it's the heme group specifically that the oxygen actually binds to. Now, this whole process is called positive cooperativity. You don't have to use that term to get the mark, but if you know it, you can put it down. It's basically just describing this idea that the binding of one molecule then makes it easier and quicker for the second molecule to bind. Right, let's look at this question. We're being asked to give the pathway that a red blood cell takes when travelling from the kidneys to the lungs. So hopefully quite an easy question, and we're able to pick up 3 marks basically just by describing the route. So we're going to start at the kidneys. So when the red blood cell leaves the kidneys, it's going to travel in the renal vein. Okay, so from the kidneys, remember it's going back towards the heart. So blood travels to the kidneys in the renal artery and then away from the kidneys in the renal vein. From the renal vein, the blood will then enter the vena cava, which takes it back into the heart. But to get that mark, you did have to say which chamber the vena cava would carry the blood into. So the vena cava will carry the blood into the right atrium. I think it's worth maybe just doing a quick picture at this point. We'll have a go at it anyway. So here's a kidney, okay? So we've got the renal vein carrying the blood back up towards the heart. The renal vein will carry the blood into the vena cava as it joins with the vena cava. and the vena cava is going to carry that blood. Here's my little heart. The vena cava is going to carry that blood back into the right atrium, okay? We've got 2 marks so far. So the third marking point, we need to get it all the way to the lungs. So it's now in the right atrium. It's then going to go into the right ventricle. So from the atrium, through the atrioventricular valves, into the right ventricle. And from the right ventricle, into the pulmonary artery, which takes it to the lungs. So from this right ventricle, it's going to go out. That's my pulmonary artery, taking it up to the lungs, where it will be oxygenated before returning back to the heart. But we're only asked for the route between the kidneys and the lungs, so we're going to stop there. Marking point wise, it's 1 mark for renal vein, 1 mark for vena cava into right atrium, and another mark for right ventricle into pulmonary artery. Right, this is a 6-mark question about tissue fluid, which I know is a topic that a lot of students struggle with. Actually, we're being asked to describe 2 things: how tissue fluid is formed and how it's returned to the circulatory system. So we've got a little diagram here, which is hopefully going to help. This is the capillary. You can see that this is the arterial end of the capillary, and this is the venous end or the venual end. These are the body cells that surround the capillary, and obviously the body cells are bathed in tissue fluid. But how is it formed? Well, the first marking point is for noting that there is a high hydrostatic pressure. So as blood comes into the capillary via the artery and the arteriole, that blood that's coming in has high hydrostatic pressure or high blood pressure. And that's what's going to lead to the formation of tissue fluid. Because that high hydrostatic pressure is going to force, now you can say water or fluid out of the capillary. So the high pressure is literally going to push out that fluid or that water through the walls of the capillary into the tissue fluid that is bathing the cells. Now, don't forget that fluid contains things like oxygen, carbon dioxide, glucose, and amino acids, as well as water. Right, so we've got the formation of tissue fluid. Another marking point you can get is to say that large proteins remain in the capillary. Yeah, the clue is here. They are too big to pass through the walls of the capillary. So only things that are small enough will be forced out over the walls due to that hydrostatic pressure. Proteins that are too large will remain, I can draw some on if you want, they'll remain in the blood in the capillary. And that's important when we start to talk about how the fluid is returned to the circulatory system. So, because large proteins remain, but remember, we are losing water from the blood, the water potential of the blood in the capillary is going to get lower. So this lowers the water potential. You can use the symbol or you can write the words. This lowers the water potential of the blood in the capillary. And that's what means the tissue fluid is going to get returned back into the blood at the venous end. Because as the blood has moved along, water has been forced out, but proteins remain. So you have a much lower water potential at the venual end of the capillary. So water will move back into the capillary. And the 6th marking point you're going to get is for saying by osmosis, or an alternative would be to say from a higher water potential to a lower water potential, which is in the capillary. Another marking point that you can get is that the excess tissue fluid, So any of that fluid that is not returned at the venule end, the excess tissue fluid will drain into the lymphatic system. Now, the lymphatic system consists of lymph vessels, and it does eventually join up with the circulatory system. So that extra fluid that drained into the lymphatic system will make its way back up towards your chest, where it will re-enter the circulatory system. So 6 marking points you needed to get all the marks. Remember, you did have to talk about the formation of the tissue fluid and the return of the tissue fluid. Hopefully that's made sense. Right, we're going to have a go at completing this table by giving 3 control measures that a student must use to reduce the risks associated with carrying and using a scalpel. Now, obviously a scalpel is a sharp blade that you use when you're completing a dissection. You may have done a heart dissection or a fish dissection, but it can be pretty sharp and there are risks associated with it. Now, interestingly, this question specifies that you've got to discuss the risks associated with carrying and using a scalpel, which we haven't really seen before on the exam. So can we think of something that we would do to make carrying the scalpel safe? Well, there were a couple of things that were allowed. One of those suggestions was that you carry the scalpel with the blade pointing down. I suppose what they mean by that is you wouldn't carry it around like that, pointing out towards other people. You would carry it maybe down by your side with the blade pointing downwards. Something else that was allowed for the same marking points, so I'll write it in the same space, is to carry the scalpel in a tray. So rather than actually handling it yourself, you put it into your metal tray where you keep your dissection instruments and you carry the tray around, which makes it a little bit safer. Now, in terms of using the scalpel, hopefully these ones are more obvious to you because this has been on the exam a few times. When you're using a scalpel, you always cut on a hard surface. So you've probably got a dissection board or a tile that you're using to cut on. You always cut on that hard surface. You should never be holding up any tissue and trying to cut it in the air because that's very dangerous, right? You always cut with the blade pointing away from your fingers. That's another thing you can say. Another answer that they would accept, if I just add on a little 4th down here, is to make sure the blade is sharp. Now you might think, hang on, that sounds like it's going to be more dangerous, but it's not. If you try and cut something with a blade that is not sharp, you tend to kind of do more of a sawing action and it's a little bit harder to do the dissection, which actually makes it more risky. It's a lot easier and safer if you're cutting with a sharp blade. The final thing that they did accept for this question, if I do a little 5 over here, was to actually disinfect the scalpel after use. Because that is still a safety precaution. You know, you shouldn't be leaving scalpels lying around that have tissue on them because that is a biohazard. So you should really disinfect the scalpel. Don't just say clean. That is not good enough. We say disinfect or sterilize the scalpel after use. So you had to have 3 answers, but remember one of them had to be about carrying the scalpel, at least one. You're not going to get 3 marks if you just talk about using the scalpel. Let's have a look at this 2-mark question taking from the genetic engineering topic, which is module 8 of AQA A-level biology. Now with genetic engineering, remember the whole point of it is to take a gene from one species and to add it to the DNA of a different species. This is actually asking us to give the name of 2 enzymes used in that process. But as well as the name, it does say describe the roles. So we have to say what those enzymes do in terms of inserting DNA fragments into plasmids. Plasmids, don't forget, are the small loops of DNA that we find in bacterial cells, and we often use them as vectors in genetic engineering. A vector means to carry the gene into the organism. So yeah, we've got two enzymes to talk about. The first type of enzyme that we're going to use is called the restriction enzyme, or to give it its full name, which you don't have to do, but it's actually called a restriction endonuclease. You can just say restriction enzyme, that's absolutely fine. Now, those enzymes are the ones that act like scissors. They break the phosphodiester bonds in the DNA. So in terms of the role, we can use these enzymes to cut the plasmid. Okay, so here's my plasmid, my loop of DNA from my bacterial cell. I'm going to use the restriction enzyme to cut the plasmid. It's going to break the phosphodiester bonds that hold the DNA loop together, and that's going to basically create a gap in that plasmid in which we can insert the new gene or the new fragment of DNA as they've described it here. Second enzyme we're going to use is DNA ligase. This is the enzyme that we think of as a bit like a glue. So DNA ligase will join the DNA fragment with the plasmid. Okay, so here's my little DNA fragment that I've already cut out of the chromosome. And remember, I've probably used the same restriction enzymes to cut out the DNA fragment as I've used to cut my plasmid. Then I can use my DNA ligase. My DNA ligase is going to stick that fragment into the plasmid, and there I have what I call a recombinant plasmid or recombinant DNA, because I've now got the original bacterial DNA and the new DNA fragment has been inserted. This question is about ADH. Now, ADH stands for antidiuretic hormone, and this hormone is involved in regulating the water content of your blood. Now, alcohol decreases the release of ADH into the blood. Can we suggest two signs or symptoms which may result from a decrease in ADH? Let's just think that through before we write anything down. If you have less ADH in your blood, then what that means is that less water is going to be reabsorbed from the nephrons of your kidneys back into your blood. So you're reabsorbing less water, which means more water will remain in your urine. So how is that going to make you feel, or what might some of the symptoms be? Well, the first thing that I think of is if you're reabsorbing less water, you're going to feel dehydrated or you're going to feel thirsty. And that can get you a marking point on its own, okay? That is a sign of a lack of ADH, possibly caused by alcohol. You feel dehydrated, you feel thirsty because your kidneys are reabsorbing less water back into your bloodstream. Now, if there's less water being reabsorbed, that means there's more water left in your urine. So the other thing we can say as a sign or a symptom, you'd experience more frequent urination, or you'd produce a larger volume of urine. And try not to just say amount, because amount, remember, is not a scientific word. Like, what does that really mean? If ever you're going to use the word amount, you can always think of something better. Like here we're talking about urine, it's a liquid, so it's the volume that's going to increase. Something else we can think about, if there's more water in your urine, then your urine would be more dilute, or you could describe that by saying it would be less concentrated, which is the same thing. Something else they actually accepted for that marking point was that urine would be lighter in colour or paler. Yeah, because that would be correct as well. If it's more dilute, if it's got more water in it, it would be a lighter colour. Right, another question on ADH, but this is looking more at how ADH actually affects your kidneys. So for 3 marks, can we describe the effect of ADH on the collecting ducts of the kidneys? Just as a side note, ADH, the hormone, actually affects both the collecting ducts and the distal convoluted tubule. If we do a quick sketch of the kidney nephron, just while we're here, this is our nephron, which is the functional unit of the kidney. You've obviously got your loop of Henle, you've got the distal convoluted tubule here, and you've got the collecting duct here. Now, ADH as a hormone actually has receptors on both the distal convoluted tubule and the collecting duct. But the question is just focusing on the collecting duct. So that's all we've got to mention. It's just something else to bear in mind. So when ADH is in our bloodstream, or there's more ADH in our bloodstream, how does it affect the collecting duct? Well, the first thing we're going to say, when ADH binds to its receptors, which are on the cells of the collecting duct, it causes the addition of water channels into the cell membranes. When I say cell membranes, I'm talking about the cells that line the collecting duct, the epithelial cells of the collecting duct. Those cells and those cell membranes are going to see the addition of water channels. Those water channels, by the way, are called aquaporins, which you probably will remember. They're like pores for water, right? Aquaporins. So by all means, feel free to say that it will cause the addition of aquaporins into the cell membranes. What's that going to do? Well, it's going to make the cells of the collecting duct more permeable to water, or we can say more water will be reabsorbed. In fact, there's another way we can say that. We can say more water will re-enter. Spelled that wrong? Spelled that wrong. We can say that more water will re-enter the blood, because that's what we mean by be reabsorbed, right? We're saying it's going back into the blood. So because we've got more channels for water, or more aquaporins in the membranes, the cells are now more permeable to water, so more water can re-enter the bloodstream. Third marking point, nice and easy, just say how that happens. This is by osmosis, okay? Because even though we're using aquaporins, that water is still moving from a higher water potential to a lower water potential, or down the water potential gradient. So we still need to say it's by osmosis. And that's it. That's how we get 3 marking points. Remember, the same is true for the distal convoluted tubule. ADH works in the exact same way there. Right, we're going to think about plants, specifically the xylem and cohesion tension theory. So this is some kind of year 12 A-level biology from mass transport in plants. Can we describe the cohesion tension theory of water transport in the xylem? Well, for this question, we always start at the top, at the leaves, because it's the evaporation of water from the leaves that actually pulls that column of water all the way up the xylem. So number 1, I'm going to start with water evaporates from the leaf cells, or you can call them mesophyll cells. Okay, the leaf mesophyll cells, or just leaf cells, was adequate for the mark. But the water is going to evaporate from those leaf cells. Instead of evaporation, you can obviously call this transpiration. In fact, transpiration is probably a better term to use, because that refers specifically to the evaporation of water from leaf cells. Transpiration. What's that going to do? Well, if you imagine that, you've now got water evaporating out of those leaf cells, it's going to lower the water potential in the leaf cells or in the mesophyll cells, okay? So you've got a lower water potential in the leaf cells and you're creating this water potential gradient. That will mean that water is pulled up the xylem. And to get a fourth marking point, we can say in a continuous, I've forgotten how to spell column, continuous column. Now, the reason we get that continuous column is because of two properties that water molecules have. So it's like, I always think of this as a bit like sucking on a straw, right? I don't know how good of an analogy that is, but if you suck on the top of the straw, the water moves up the straw in a continuous column. And that's because water molecules are both cohesive and adhesive. So that's what we need to talk about next. So water molecules are cohesive, which basically means they stick to each other, and they form these hydrogen bonds, don't they? These intermolecular forces between each other, so they kind of stick to each other. And water molecules are also adhesive with the xylem walls. So not only are they sticking to each other, but they're also sticking to the walls of the xylem. Now that cohesion and adhesion helps the water to move up in a continuous column. So it's moving up from the roots up towards the leaf cells, because remember, the leaf cells have a lower water potential. So the water's moving down that water potential gradient, but in a continuous column, it gets pulled up because the water molecules are both cohesive and adhesive. This creates a tension in the xylem, doesn't it? Which is why it's called the cohesion tension theory. Let's describe how mRNA or messenger RNA is produced in a plant cell. Now, this question is basically asking you to describe transcription, which we have done in a previous video, but there were quite a lot of you that were asking me to go over this topic again. I know it's one that we do find quite difficult. So this is worded slightly differently, but it is basically asking us to describe transcription, which is the process of making messenger RNA, right? So the first thing we're going to say is the DNA strands are separated. But to make sure we get that marking point, we need to say either that the hydrogen bonds are broken, or we need to name the enzyme that does it. So this is done by DNA helicase. Okay? So don't just say the DNA is unzipped. Yeah, a lot of students say that. That is not scientific. What's really happening is that the helicase enzyme is moving along the DNA. It's breaking the hydrogen bonds to separate the two strands. So that is the best way of saying it. Now, only one strand will act as a template, which is different to the process of DNA replication, where both strands act as templates. Here, just one strand acts as a template to make the messenger RNA. Okay, now we've got our template strand. RNA nucleotides line up to the exposed bases. And you can actually get another marking point for describing the base pair rule. So how do those nucleotides line up? Well, if you've got a T on the DNA, that would pair with an A on the RNA. If you've got an A on the DNA, that would pair with a U on the RNA. G on the DNA would pair with a C, and a C on the DNA would pair with a G. So you can actually get a marking point for giving those complementary base pairs. Now, once they've lined up according to the base pair rule, the next thing that's going to happen is RNA polymerase. I'm just going to underline that. In fact, I'll underline that as well. This is our second important enzyme. RNA polymerase will join the RNA nucleotides together. I'm going to put in brackets by making phosphodiester bonds. That wasn't actually needed for this mark scheme, but it has been on previous mark schemes. So if you can remember the name of those bonds, it's definitely better to include it. Right, so RNA polymerase has moved along. It's joined one nucleotide to the next by making phosphodiester bonds, but we're not going to stop there. I mean, we have actually got 5 marks, but there's a couple more things that we can always say. That initial RNA is called pre-mRNA. And the reason we call it pre-mRNA to begin with is because it still contains introns. It still contains those non-coding regions, right? Which we do need to remove before translation. So there was a 7th marking point, which was the pre-mRNA needs to be spliced or the introns need to be removed. And that's it, that's transcription. Once we've got our mature messenger RNA, as we call it, it's going to leave the nucleus through a nuclear pore, it's going to go to the ribosomes where it's going to be translated and we're going to make the polypeptide. It's one of those stories, you've just got to learn it. Right, we're going to have a go at defining 2 very important terms in biology. So the first word we've got is genome and the second term is proteome, and if we can give 2 decent definitions, we can get 2 marks. So what is the genome? Well, the genome is all of the genes or all of the DNA in a cell, or you can say in an organism. Okay, nice and easy. It's all of the genes, all of the DNA inside a cell or inside the organism. So what's the proteome then, and how is it different? Well, as we know, genes code for proteins because the order of bases in the DNA determines the order of amino acids in a protein. And obviously proteome is linked to the proteins coded for by the genome. So to get the mark for proteome, we can say the range of proteins that can be produced by a cell or an organism. Equally, if you want to do it slightly differently, you can say the range of proteins coded for by the genes or the DNA or even the genome. Okay, but you need to convey the idea that it's a range. It's all of the different proteins that can be made in the cell or all of the different proteins that are coded for by the genes or the DNA. Right, with this question, they're asking us to describe how one amino acid is added to a polypeptide that is being formed at a ribosome. If you hadn't noticed, this is basically asking us about translation. Now, translation is the second stage of protein synthesis. It comes after transcription, and we have done a video on the whole of translation before. So go back and watch it if you want to just recap everything about translation. This question is only 4 marks, and it's not actually asking for the whole of translation. It's just asking us to explain how one amino acid can be added to the growing polypeptide. So let's just start there. First thing to say is it's transfer RNAs that bring a specific amino acid to the ribosome. Okay, so they're called transfer RNAs because they carry and they transfer these amino acids. But remember, each tRNA can only carry one specific amino acid. They can't just bring any old amino acid in. And the amino acid has to be able to bind to the binding site, right? So it's important we say that the transfer RNA brings a specific amino acid to the ribosomes. Okay, what's going to happen next? Well, the way it works is the tRNA anticodon binds to the mRNA codon. Let me just do a quick picture to illustrate that. So here's my mRNA. Now remember, my mRNA was made in transcription. It's a copy of the DNA code. It goes to the ribosome. Now, 3 bases on mRNA is called a codon. And let's imagine that these 3 bases are A, G, okay? Now the tRNA that has the complementary anticodon will be able to bind to that codon. So here's my tRNA. That's a bit of a rubbish diagram, but go with it. The important bit is that the anticodon will be complementary to the codon. So if the codon is AAG on the mRNA, then the anticodon would be UUC on the tRNA. Remember, there's no thymine in RNA. It's uracil in place of thymine. Yeah, my tRNA looks a little bit dodgy there, but the reason I drew it like that is because we often describe tRNA as having a clover leaf shape. So that's my best attempt. Right, once the tRNA has brought the specific amino acid and its anticodon has bound to its codon on the mRNA, what happens next is that amino acid will be joined to the previous amino acid using a peptide bond. So naming the bond that forms between those 2 amino acids can get you marking point 3. Equally, if you don't name the bond, but you say it's a condensation reaction, yeah, because you're forming a bond and you're also forming water. If you say it's a condensation reaction, you could also get marking point 3. Finally, making that bond uses ATP. Does require energy. That's how we get marking point 4. Now I'm just going to stop there because all it asked me to do was describe how one amino acid is added to the growing polypeptide. So I don't need to continue and say that the process is repeated or the ribosome moves along, because I've now added one amino acid to my growing polypeptide using ATP in a condensation reaction, making a peptide bond. Right, we're going to do this question about aerobic respiration, specifically stage 2 of aerobic respiration, which is the link reaction. And I don't think we've done a video on this stage before. So we're going to try and describe how acetyl coenzyme A is formed in the link reaction. 2 marks. Now this equation wasn't given to students in the exam, but I've just written it down to kind of help me explain exactly what goes on in the link reaction. Now the link reaction follows on from glycolysis. Now you'll remember that in glycolysis we've made pyruvate. In fact, we make 2 molecules of pyruvate from each glucose. Pyruvate is then actively transported into the mitochondria, into the matrix, where we get the link reaction. So what happens to the pyruvate? Well, if I'm going to word this out with a written response, I'm going to say pyruvate is decarboxylated, which basically means it loses carbon dioxide. And you can see that's why we've formed carbon dioxide as one of the products, because pyruvate loses carbon dioxide or is decarboxylated. Second thing we're going to say, pyruvate is oxidized, or we can say dehydrogenated, Okay, remember oxidation is loss, and in this case, oxidation is loss of hydrogen. And another term we can use for that is dehydrogenation, which just means to lose hydrogen. Now, the pyruvate loses hydrogen, and you can see that that hydrogen is accepted by a coenzyme called NAD. So we needed NAD to accept the hydrogen that comes from pyruvate. So NAD is getting reduced while pyruvate is being oxidized. Now, another marking point that's available to you is coenzyme A is added. Because at the end, once we have decarboxylated and dehydrogenated the pyruvate, we have the acetyl group. And the acetyl group then joins with the coenzyme A to form acetyl coenzyme A. And that's the end of the link reaction. As you guys may know, acetyl coenzyme A can then go on to the third stage of aerobic respiration, which is the Krebs cycle, also happening in the matrix. So, only a 2-mark question. The link reaction is quite a simple stage, not too much to remember, but you've basically got pyruvate being decarboxylated, being oxidized, and then you've got the addition of coenzyme A. Right, with this question, we've got 2 definitions to think about. Now, both of these terms relate to the genetic code. We know that the genetic code is both degenerate and non-overlapping. What do these terms actually mean? Well, the first term, which was degenerate, is that more than one triplet, which remember is a set of 3 bases in DNA, more than one triplet can code for the same amino acid. Now, it would also be totally acceptable for you to say more than one codon can code for the same amino acid. We quite often use the word triplet and codon interchangeably, but what the difference actually is a triplet is a set of 3 bases in DNA that codes for an amino acid, and a codon is a set of 3 bases in mRNA that codes for an amino acid. But in terms of the word degenerate, you can say either. It's just this idea that you can have more than one triplet or codon that codes for the same amino acid. For example, we use an example to understand it. All of these codons, all 4 of those codons all code for the same amino acid. They all code for valine. So I think that helps to illustrate the point. It also means that sometimes if you get a mutation in the DNA or the mRNA, it doesn't have an effect on the final protein. For example, if this was substituted for an A, you change the base sequence, which is a mutation, you substitute the G for an A, it actually doesn't code for a different amino acid. It's still the same amino acid, so the protein won't be affected. Right, non-overlapping. The genetic code is non-overlapping. What does that mean? Each base is only part of one triplet or one codon. And again, we can do an example for that. If you think about your DNA, let's say you've got a series of bases that looks like this. AGT is the first triplet, CCG is the next triplet, and ATC is the next triplet. So each base is only part of one triplet. There's no overlapping. You don't get a base that's the end of one triplet and the start of the next. There's no overlapping in the bases. So that's what non-overlapping means. The other 2 terms, by the way, that you need to know for the genetic code are universal and triplet code. So triplet code means that 3 bases code for one amino acid, which is nice and easy. But universal means that the same triplets code for the same amino acids in all living things, which is really good evidence for evolution.